Hello, my name is Connor Howgard, and I'm a senior biology major at Concordia College, and I am from Detroit Lakes, Minnesota. My project was looking at how the introduction of zebra mussels changes the zooplankton populations within lakes and how that can ultimately impact the fisheries of those lakes. Overall, zebra mussels, or Dracaena polymorpha, is an invasive aquatic species that is native to the Caspian region in Russia and has been increasingly introduced to freshwater ecosystems throughout the world in Europe and North America especially. In North America, we've seen a lot of infestation done in the St. Lawrence Seaway, Great Lakes region, and they've slowly made their way farther inland and were first identified in the Duluth Harbor in 1989. This project was done in the Pelican River Watershed District, which is the lakes managed around Detroit Lakes, and is called PRWD for short. The PRWD generally found its first case of zebra mussel infestation in Lake Melissa in 2014, and then subsequent infestations on Lakes Sally and Detroit in 2016. The Floyd Lakes, that is the topic of this presentation, in 2018, and then Long Lake in 2020. Zebra mussels are normally classified as what is called an ecosystem engineer, which are organisms that will significantly alter the ecosystems that they are introduced by outcompeting the native mussel populations. And for zebra mussels, this is done through increasing the filtration of the water column at much higher rates than the native population that will drastically increase the water clarity, bringing the littoral zones of the lake, or the area of the lake where the light is able to penetrate down to the bottom deeper, that will increase sunlight penetration and increase aquatic vegetation within the lake of both native species and non-native species such as flowering rush. These underlying community changes are important to consider as it can alter the food web, with the zebra mussels outcompeting the native population's niche, and then increasing filtration, which reduces the overall nutrient levels that the primary consumers rely on, such as phosphorus and nitrogen. Another unique thing about zebra mussels is their ability to selectively filter algae that in the Great Lakes has contributed to dangerous blue-green algae blooms that are not necessarily seen yet in the Pelican River Watershed District or other lakes kind of in the area that are more inland and smaller that have had zebra mussel infestations for quite a long time. Zebra mussels also play a large economic impact, as we kind of see in the picture. I'm holding basically a snail, and then on the snail shell, we see many different individual organisms of zebra mussels. And this is because they secrete a substance that is basically like cement and cements them to basically anything hard in the lake. So this spells a lot of problems for lakes with a lot of rocks, or anything that someone puts in the lake that's hard, like a dock, a lift, a boat, or in this case, other organisms. The purpose of this project was to look at how zebra mussels impact zooplankton populations, and overall zooplankton is a broad classification of species that are primary consumers within the lake. Zooplankton feed on algae, phytoplankton, some other zooplankton, as well as just nutrients and any form of vegetation in the lake, and are preyed upon by a lot of fish fry or year one developing fish in the lake, and so as a major source for their growth and development. With zooplankton being filtered out by native mussels as well as zooplankton, we do possibly see, with increased filtration of zebra mussels or of zooplankton, a lot of environmental destabilization because of this, and a reduction of zooplankton can alter the fish populations, as the younger fish fry will not have the nutrients to grow and develop quite as much. And this impact that zebra mussels can have on the fisheries is of great interest, especially with subsistence fisheries or areas like Lakes Country in Minnesota that rely heavily on the economic power of the lakes to support hotels, bait shops, fishing excursions, and just the whole community that surrounds some of these lakes, and can definitely also impact indigenous and poor residents that use either traditional methods for some of their livelihood, or people who use fish as a primary source of food and protein. Overall, we could fix this by stocking at the lakes, but this is expensive and does require some kind of government support to do. So this kind of trophic cascade that we could see with the introduction of zebra mussels has a potential to include humans and contribute to increased suffering of some of these people. Overall, the goals of this project was to taxonomically group and to quantify zooplankton in the Floyd Lake chains, which was Big Floyd, Little Floyd, and North Floyd, which were our zebra mussel infested lakes, and compare them to a zebra mussel non-infested lake of Long Lake, which is where our Concordia Field Station is, within the Pelican River Watershed District, and expand the project to study the effects on fishing populations through trophic cascades. My hypothesis overall was that lakes with zebra mussel infestations will see a decrease in density of the zooplankton populations, and this will increase stress on the subsistence fisheries due to the trophic changes. Overall, samples were collected between June and September of 2019, and this was before Long Lake was found to be zebra mussel infested. In 2020, Long Lake was actually found to be zebra mussel infested. And it followed at 3 distinct sites following Minnesota DNR standard protocol for zooplankton collection. And that basically used an 80 micrometer net that you see me holding in that picture that was lowered down through the water column until the collection container at the very bottom was about 2 feet from the bottom of the lake. And then the samples were slowly raised at about a half a meter per second throughout the water column. And then once the net was out of the water, we rinsed the zooplankton down into the collection container with clean distilled water, and then narcotized the sample with Sprite. And what the Sprite did was basically the carbon dioxide and the sugars in the Sprite would make the zooplankton high, basically, and kind of put them in a more relaxed state. So when we would ultimately preserve them with 95% ethanol, they would be easier to identify taxonomically later in the lab. So we brought the samples back to the lab, and using a dissection microscope, we sorted them and counted them based on taxonomic groupings of ratio cladoceran to copepods, percent cladocerans, and copepods, as well as any other organisms that we found down to the species level. And then data such as total density and percent grouping were found. The total number of organisms were calculated, and statistical analysis was done using RStudio, with my dependent variables being ratio of cladoceran to copepods and density. The independent variable that we looked at was the lakes that were sampled, and then the data from the fishing part of the project was collected using a literature review, as well as interviews from a fishing guide and Mille Lacs Lake Resort. The results are shown here, with the graph on the left being the density on the y-axis and the lake on the x-axis. And what we see is that Big Floyd had the lowest density of zooplankton with 8 organisms per liter. Little Floyd had a median density of 14.8 organisms per liter. Long had 18.8 organisms per liter, and North Floyd had 19.4 organisms per liter. And the numbers that you, or the letters you see on top of the graphs with the A, B, C, and BC, that shows significance within the data. So if the number or the letter above each lake is different from another lake, that means that the data is significant. But if it is same, it is not significant. So what we see is Big Floyd shows significance in the difference of zooplankton between Little Floyd, Long, and North Floyd. Little Floyd is significant between Big Floyd and Long, but not North Floyd. Long shows significance between Big Floyd and Little Floyd, but not North Floyd. And North Floyd simply shows significance between Big Floyd. Then on the graph on the right, we see the basically log of the ratio of cladocerans to copepods varying on lake. And this basically means a number of, or median number of 1 would be equal number of cladocerans to copepods. A number lower than 1 would be a greater number of cladocera to copepods, and a number higher than 1 would be more cladocera. And what we see here is that Big Floyd had a ratio of 0.12, Little Floyd had a ratio of 0.94, Long had a ratio of 0.39, and North Floyd had a ratio of 0.16, with the letters on top of the graphs indicating that we saw significance between every single lake we studied. The results do indicate that there are higher densities of zooplankton in Long Lake as compared to Big Floyd, with a p-value less than 0.001 for the ratio of cladocerans to copepods as well as density. And this does point to zebra mussels decreasing zooplankton densities, since Long Lake is a non-infested lake and Big Floyd was one of our infested lakes. North Floyd and Little Floyd both had very, very similar densities to Long Lake, with North Floyd being 19.4, Little Floyd being 14.8, and then Long being 18.8. We did see ratios of cladocerans to copepods in both North Floyd and Little Floyd being higher, with Little Floyd being basically 1, the same amount of cladocerans to copepods, and North Floyd having a higher rate of cladocerans. This does appear that zebra mussels do not impact zooplankton densities in North Floyd and Little Floyd. However, the ecologies of both North Floyd and Little Floyd differ significantly from the lakes of Big Floyd and Long Lake. And this has to do a lot with the trophic levels. So Long has a trophic level of 41, while Big Floyd has a trophic level of 43. And this basically both means that they're mesotrophic lakes, which what a mesotrophic lake is it is a lake that has basically a median number of nutrients. So a median number of phosphates, a median number of nitrates. And this is kind of just the standard lake. It's the good lake we want. It's just a standard lake. While both North Floyd and Little Floyd have trophic levels that are higher in the 51. And that means that the lake is eutrophic, which means that it is high in nutrients. And so this means that it has elevated levels of nitrogens and phosphates. And we can see that in the picture there. So the sample in the middle, that is one of the samples that I took in July of that in 2019. And that was a Big Floyd, the clear one in the center. And then the ones on each side, we have North Floyd on the left and then Little Floyd on the right. And you can see just kind of the differences in the overall water quality. I will say though, that these samples do kind of make both Little Floyd and North Floyd to be really, really dirty lakes. They're actually still pretty nice lakes. But we kind of see just the general increased amount of turbidity, or kind of floating particulate matter in the lakes, that is one of the things that will bring up trophic levels. So kind of from that, we do see lakes with the same trophic level have significant decreases in zooplankton densities following zebra mussel infestations. And the difference between densities of cladocerans is of great importance, because those are the Daphnia species. And those are, Dr. Marco always described them basically as fat, juicy, and slow. And they're the Daphnia species that we see. And this is really good for the fish fry because they lack some of the predatory aversion that the copepods do, that are a little bit more streamlined, have a bit kind of more tail-like structure, that are able to kind of fight against predation as much. And this also would go into kind of some zebra mussel filtration, as zebra mussel would kind of have that negative sucking force that copepods would be able to better go fight against, rather than the Daphnia species. And we saw that within the ratio, where our ratio results, where Big Floyd had extremely low levels of Daphnia species and cladocerans. And this is one kind of major thing that we are looking at, and how that decrease of cladocerans points to Daphnia being filtered out at higher rates than copepods in lakes that are infested with zebra mussels and taking down their populations at a higher rate than taking down overall zooplankton populations. From the results of kind of the fishing, I did a survey with Brad Labs, who was a fishing guide in the Detroit Lakes area, and he generally says that weed beds have been pushed farther, about 10 feet deeper into the lake, from about 12 feet to 22 feet. And this does point to kind of the standard things we know about zebra mussels, with increasing clarity, allowing littoral zones to be pushing deeper into the lake. And this kind of changes where fish are, as a lot of fishing is done on those weed beds or on the border of the weed beds. In Mille Lacs Lake especially, we saw an overall shift in size of walleye being a little bit bigger, actually, at least being caught walleye being bigger. And I think part of this is because we see a lot of these larger walleye being caught as they're the apex predator, while a study done by Gretchen Hansen's lab at the U of M found that the first year walleye, or the walleye that are basically in their first year of life, are a little bit smaller than lakes that are infested compared to lakes that are non-infested, even though they do kind of catch back up later on. And this could be one part, is that the overall kind of trophic levels of lake are being switched, and we're not seeing so much population of walleye in kind of that normal range, and we're seeing a lot more in the higher population, and they're kind of possibly doing some predation of some of the younger walleye. And this overall does pose a problem for those who rely on fishery for subsistence, as we're seeing basically fish going deeper and the use of boats and expensive tracking a lot more frequently now. And for poor populations or people who are used to a more traditional way of fishing, such as indigenous populations, they're going to be losing some of that ability to catch fish at the rates that they're used to, along with some of these kind of old honey holes in the lake being completely gone now, or just changed locations due to the changes in the littoral zones, water clarity, and the overall level of aquatic vegetation. So overall, these ecological changes from zebra mussels, or possibly due to zebra mussels, will require more of an active management that is imperative to curb the lake and fishery from collapse, and places a large importance on a lot of government organizations such as the Minnesota DNR and watersheds to basically fight against the current invasive species that we have, and also protect against further infestations of other invasive species. Overall, the study was conducted in order to quantify zooplankton densities between lakes infested with zebra mussels and non-infested with zebra mussels, and see how this will change zooplankton as well as the trophic web that can affect fishing. The data shows that between mesotrophic lakes, zooplankton densities do decrease following zebra mussel infestation, while eutrophic lakes saw generally a similar or greater density of zebra mussels, or of zooplankton. However, more research will need to be done to see if the nutrient level in the lakes was a confounding variable, as the nutrients could provide basically extra source for the zebra or the zooplankton to use and to have a more robust and large population. The fisheries in the lake really haven't seen a decrease in the number of fish caught. However, the increases in water clarity has pushed aquatic vegetation beds into different regions, and the fish populations as well as we have seen how walleye have stunted growth for the year. Overall, for continued success of the fishery and the people who rely on it, organizations like the Minnesota DNR, other government agencies such as the Pelican River Watershed District should closely monitor the food webs and fishing populations and take a more active role on stocking fish and just monitoring the changes of the lake for continued ecological success after a zebra mussel infestation. Overall, I would like to thank the Pelican River Watershed District for sponsoring this research and providing the funding and just use of equipment that I needed for the project. I would also like to thank Dr. Marco for serving as my primary and first advisor on this. She just gave a wealth of information on lake ecology and zooplankton identification that I would have never been able to do or dream of without her help. along with Dr. Jennifer Sweatman for being able to kind of pick the baton up on this project and helping me push it in a lot of new directions, as well as just bringing it towards the finish line. Thank you.